Damjanovic. She's from Johns Hopkins University. She received her PhD in physics from University of Illinois at Urbana-Champaign with Professor Klaus Schulten. Then she started working on photosynthetic light harvesting, and she kept working on this during her postdoc at UC Berkeley. Then she moved to Johns Hopkins University, working on internal ionizable groups through molecular dynamics simulation. She is still working in Johns Hopkins, and especially focusing on constant pH simulations and study of ion channels. The talk of today will be pH and other determinants of conductance of the bacterial voltage-gated sodium channel. Thank you, Anna, for being here.
Yeah, thank you for having me here. So, hi, everybody. So let me share screen. So can you see everything and can you hear me well?
Good, yes.
Okay, very good. So voltage-gated ion channels are exciting because they contribute to the action potential. So they respond to changes in the membrane potential. So when a threshold is reached, they can open, and that lets sodium ions into the cell. So that results in the changes in the membrane potential. So then potassium channels open, and after a while, both channels inactivate, and the resting state is achieved. Sorry, I'm trying to, okay, able to move. So, sodium channel, voltage-gated sodium channels are tetramers. So they have the voltage sensing domain and the pore domain, which obviously contains here the pore. So the channel can exist in open, closed, and in inactivated states. So this is the voltage sensing domain, which has some ionizable residues that can respond to the change of the membrane potential. So they can move, so the whole voltage sensing domain can move and can pull on this linker here. And that will open the gate here at the bottom. I don't know if you can you see my mouse? Can you let me know?
Yes, it's visible.
You can see my mouse. Okay, very good. Yeah, so it will open the gate, and that will allow sodium ions to pass through. And this is the pore domain. So of course, in addition to the gate, which can be either closed or open, there's the selectivity filter with select sodium ions. So this is what it looks like in eukaryotic channels. So it has OSC, glue, alanine, and lysine residues. And then in bacterial channels, it has 4 glue residues. So bacterial channels are fully symmetric. So pH actually influences these mammalian sodium channels. So it is known that reduction of extracellular pH affects multiple functions. So it decreases maximum conductance with a pK of about 6. And then it also affects activation and inactivation of the channel. So a lot of experiments have been performed, and multiple residues, they indicate that multiple residues play a role. So the important ones are protonation of residues in the selectivity filter and protonation of other miscellaneous residues. However, the interpretation of these experiments is, in terms of mechanism, is not straightforward. Now, this is just some interesting facts, actually, that with breathing, you affect your blood pH, and that in turn can influence the neuronal excitability. So, for example, there were experiments in so if you hyperventilate, it results in reduction of CO2 levels and in more alkaline pH, and that in fact, in turn, induces hyperexcitability. While hypoventilation results in hypoexcitability. And the mechanism for this involves pH-sensitive membrane proteins, including our voltage-gated sodium channels. And this is just to show some pH-sensitive proteins in neurons, so there are a lot of them. And to go back to our sodium channels and the selectivity filter. So this is the eukaryotic sodium channel selectivity, and the bacterial, I already showed them. So actually for both of them, lowering of pH results in decreased conductance and alters activation and inactivation. Then there are calcium channels that actually have selectivity filters similar to the bacterial channels. So it has have the 4 glue residues. And there were experiments that showed there are two conductive states at pH 7.4, so at 7.4, so at physiological pH, and that actually mutating one of these glues to glutamine abolishes the high conductance state. So the interpretation is that higher conductance states correspond to more or deprotonated state of the selectivity filter, and just protonation of one will result in lower conductance state. So the question is then for our channel, so we studied this bacterial channel NAV-MS. So what is the protonation state of this tetrad residue? So this is the selectivity filter. So obviously it's going to impact the conductance. And most of the molecular dynamic simulations that were performed with this channel considered this state to be fully charged. There were some with that considered different protonation states. So this was a first step in our study, is to perform simulations with different protonation states of this glue tetrad. So I kind of just want you to remember here that S0 we are denoting the states that's fully deprotonated. So you can think of it as 0 protons. And then S1 is singly protonated, so 1 proton. And then we have S2 states and S3 states with 3 protons. So the simulations showed right away that there are actually different conformations of the selectivity filter depending on the protonation states. So for fully deprotonated, so zero protonated, S0, all of the glue residues are pointing up. So this is showing the Z coordinate, average Z coordinate in the simulations. So they're mostly in one Z value. So there is some fluctuations, but not too much. So they cluster here. And then when you protonate one, it goes further down into the channel. So you can see this here. So this is for S1, the Z values. So you see the protonated one has lower Z value. And then for S2 state, so these are 3 different S2 states. Again, the protonated one are buried further inside the channel. So of course, when we're doing PK calculations, we have to take into account these conformational changes. The other thing that's also immediately noticeable, the difference in the number of bound ions for simulations with different protonation state. So with the S0, there are 2 to 3 ions, mostly 2, sometimes 3. And then for S1, there's about mostly 1 ion, and then sometimes 2. And then for S2 state, it's mostly 0 or sometimes 1. And for S3, we even observed a chloride ion moving in. So obviously the protonation states influences the number of bound ions. So our work is consistent with the previous MD work that investigated this. So what hasn't been investigated is actually these PK values based on molecular dynamic simulations. So we do this through constant pH simulation, through enveloping distribution sampling and Hamiltonian replica exchange. So this is the method that we worked on and developed. So it is based on the enveloping distribution sampling method by Christ and Van Gusteren. So it is a method to calculate free energy differences from a single simulation. So let's say you have states A and B, you can construct a reference Hamiltonian for which the partition function of the reference Hamiltonian is the sum of partition functions for states A and B. And now the problem with this method is that if you have a huge energy barrier, you're going to be stuck in one state. And that's generally a problem in constant pH simulations in explicit solvent because of water molecules. There's all organized themselves to favor a particular protonation state. There is a huge barrier to actually sample the other state. So sampling of protonation states is a huge problem if you're looking into methods for constant pH simulations in explicit solvent. Okay, so I made the digression here, but Christ and van Gunsteren introduced this smoothness parameters, S, in the Hamiltonian, where they have a smoothed Hamiltonian where they can actually easily transition between states. Now, the problem is that this smoothed Hamiltonian samples non-physical ensemble. So what we did is we introduced a Hamiltonian replica exchange where you perform simultaneously simulations with different smoothness values. So you have your physical Hamiltonian that's non-smoothed, but the transitions are hard. And you have the smoothed Hamiltonian, which is non-physical, but you can easily transition. So in these constant pH simulations, then you can sample at the physical Hamiltonian and still get transitions. You just have to have a lot of simulations. And then we introduced a pH-dependent offset so that we can do constant pH simulations. And in this case, with pH, you can favor different states depending on the pH. And then we introduced a two-dimensional version of this method with the two-dimensional replica exchange, where we have exchange in pH on one hand, which is again going to favor different protonation states. And we also have replica exchange in smoothness parameter, which is going to facilitate transitions between different states. So this method results in faster convergence and smoother titration curves. So, so this work actually is the first application of this method to a big protein system. So when it was developed, the method was developed, we applied it on small systems. So this is a big system. So we used 50 replicas, 10 pH values, and 5 smoothness values. And you have to actually, in this method, you have to explicitly include all of the states in your Hamiltonian, right, in this reference Hamiltonian. So this just here, cartoon is showing just 2 states, so protonated and deprotonated. However, we're looking at this 4 residues here, right, in this selectivity filter. So there are 2 to the 4 states really that are feasible. So because the 3 times protonated and 4 times protonated are non-conductive, we excluded them from the simulation to reduce the number of states because these simulations are really huge. So we included 1 deprotonated states, and then there are 4 singly protonated where each one of the 4 glue residues can be protonated. And there are 6 different combinations for doubly protonated. So that's a total of 11 states. So these were really, really huge simulations. And then when we got the pK values, they were all over the place. So then we tried to make sense of them. And first thing we noticed is that there's a huge hysteresis in calculated values depending whether you start the simulation. So you equilibrate the system, let's say in S0 state, you equilibrate it in S1 state, and you get vastly different values, right? So depending how you equilibrated the system and what you use for your initial structure for constant pH simulations, you get different results. So we then repeated this calculation with free energy perturbation. because that's an established method, and we wanted to make sure we're getting the same results. And indeed, we actually got the same results, very, very, very similar values. So you can see here in this pink and red pK value with 0 ions in the channel, and with the EDS, and with free energy perturbation. So they're very close to each other. And then, so this is started from the S1 state. The blue ones are started from the S0 state. So they're very, very close to each other. And then we noticed that there is a dependence of the pK values on the number of ions that's in the channel. So we have this for S0 to S1 transition, and then from S1 to S2 transitions. So, so free energy perturbation were actually much faster than our EDS. So we decided to continue then with the free energy perturbation. So we were able to run them longer. And this is what the results look like after, I think, 10 nanoseconds of simulation. So those simulations actually involved 11 replicas. We were doing Hamiltonian replica exchange to get these numbers. So, the hysteresis was much reduced, and you can immediately tell this dependence on the number of ions on the pK value. So to look at what this means, so this means that if there are 0 ions in the channel, that protonated state, so this protonated state is definitely populated. And if you look in the S1 to S2 transition, that even the deprotonated state can be populated. With 1 ion in the channel, you definitely also get protonated, singly protonated, and you can also get doubly protonated. With 2 ions in the channel, you can get both singly and doubly a little bit, and 0, 0, 1, and 2. And then with 3, you mostly get 0, deprotonated, fully deprotonated, so as a 0 state. So in conclusion, at pH 7.4, the physiological pH, which we're looking at here, both all deprotonated singly and doubly protonated states are feasible. And then we more recently performed much longer free energy perturbation, so for 300 nanoseconds. And we obtained convergence in these simulations, so with a pK about 6.7. And this is here I'm showing results. So there are experimental results on several bacterial channels, unfortunately not on the exact one that we are studying, but they all have a very similar selectivity filter, and there's definitely a pH dependence on the current, maximal current, and with a pK of about 7.6. Again, this number varies a little bit for different channels, but they're all of them are very high. So in conclusion on this part, the protonation states in the selectivity filter get modulated by number of ions in the channel. So in a way, there is a competition between ions and protons. And then 3 different states, so fully charged, deprotonated, singly protonated, and doubly protonated are possible. So next step, we wanted to look at the conductance for different protonation states. So I'm going to show you the movie here. Hopefully it works. So let's see. So you can see ions moving through. And actually, it's kind of a knock-on mechanism. So one of them comes and knocks the other one down. So, it's kind of a nice movie, but it was actually very hard to get it. Actually, let me get back. So the reason is that when you start the simulations from the crystal structure, the gate here, it actually closes. And this didn't happen just for us. This also happened for other groups who were doing these simulations. So one of the groups introduced some mutations in the hydrophobic residues here. So it mutated them to alanine to make a little bit a wider gate. So that's what we did. And then we were actually able to observe ion conduction events. Now what happened is that we obtained values that are kind of very different. So we got some really small values, and then we got larger values. And then initially we only had 3 simulations. Then we decided to submit 17 more. So we had total of 20 simulations. And they're about 500 nanosecond long molecular dynamics simulations. So we get, again, a distribution of values. With a lot of them with just very poor conductance and some higher conductance. And the experimental value is about 33. So they were all smaller than 33. So we were wondering what's going on here, and then why are these conductances so different? So we investigated a bunch of factors, and after a while we plotted these pore radius. And that turned out to correlate really well with conductance. So you can see here, this is the radius of the pore. This is Z direction along the channel. So this is selectivity filter. This is the gate. And in dark, the darkest line here, so the lines are colored by conductance. So you can immediately see the darkest one has the highest conductance, right? So the gate here actually is widest, right? And then the lighter one, which have smaller conductance, have a smaller gate. And this actually does make sense, really does make sense. There's also on this plot is a correlation between conductance and gate radius. So really gate radius correlates best with conductance. We looked at the selectivity filter, in some cases, if we looked at all atoms, it wasn't that well correlated, but when we looked at the backbone atoms only, there was some correlation. So, so what happened actually is that the gate further shrinks a little bit from the PDB structure. So then we decided to actually constrain the gate to the PDB value. So we didn't allow those residues to move further in. They were able to fluctuate, but not go further in. And then looked at conductance. So there was a dramatic really change in conductance. So if you look at the average values, so these are averages over three sets of 500 nanosecond long simulations, and for different voltage values. You can see that the conductance here in black are the gate restraint simulations. So it's much higher and much closer to experimental value. While those, of course, where the gate collapsed are much, so these are unrestrained. The conductance is much smaller. So here in this plot, you can see the radius of the gate shrinks, and this is what it is when it's constrained. And of course, we're also showing the potential of mean force for this. So then we checked whether the selectivity filter is affected by these gate restraints. So we were comparing sodium ion binding patterns in the selectivity filter, so how it interacts with other residues. So with no restraints and gate restraints, they look really almost identical. So the selectivity filter is not affected by these gate restraints. So then we proceeded to look at how protonation impacts the conductance. So I'll show more movies here. So this is the old movie with the fully deprotonated selectivity filter. And this is the new movie with a singly protonated residue. So you can immediately see that there are less ions in, and they're actually moving slower through the channel. Right, so I'll kind of let them finish. So then we wanted to quantify this. Oops. So these are the results again for conductance for different protonation states. So in red is the fully deprotonated, so that's the highest conductance. So these are actually the numbers that you've already seen before. And then these are new numbers for singly protonated is blue, and then these are doubly protonated. And gate, the radius is not affected a lot. There is some change here, actually, for the selectivity filter, and this is potential mean force. So the conductance really decreases with each proton bound, and this is kind of what we really expected. It's a because there were all experiments that showed the decreased conductance with pH, with the lowering of pH. We also looked at the effect of lipids because ion channels can actually get affected by lipid composition. So we also see some change in conductance with when we look at neutral lipids in red and charged lipids in blue. So actually not charged, but really mixed. So we looked at the proportion, the number of charged lipids in the actual experimental system, and used similar lipids, and definitely similar proportion of charged lipids. So you can see that there is slightly higher conductance, although the errors are pretty big. So the charged system has the charged lipids also increase conductance, and the pore is not really affected much by this. And then we also noticed we did a lot of simulations, actually a total of 170 different simulations. I'm not showing all of them, and they were all 500 nanoseconds long. So we looked at and they were performed with different voltages. So we looked at we noticed that there is sometimes there is higher voltage. Actually, higher voltage will result in higher conductance. So we looked at the correlation, and we plot here also the radius. So these in this plot, the coloring is by voltage, right? So the black is so this is lower voltage, this is higher voltage. And you can look at the radius of the selectivity filter. So it's smaller for lower voltages than for higher voltages. And this trend, it's hard to follow all the colors, but I can tell you that there's a trend that higher voltage has a higher rate, slightly higher, so that we're talking about 0.2 angstroms here. So slightly higher radius, but the conductance is also slightly higher. So one has to be careful when doing those simulations at higher voltages, because they can slightly bias the simulations and the conductance values. And then we also looked at the pK values. So this actually I already showed before. This was done with charged lipids. With neutral lipids, we are getting similar pK values. They are less converged in this case. And we also looked at the pK values under different voltages. So those are voltages in red. So there are slightly higher pK values when we turn on the voltage. Yeah, so that's basically our most recent work that has recently been submitted to Biophysical Journal. Hasn't been published yet, so it's being reviewed. So these are the conclusions. So gate restraints increase the conductance. So by gate restraints, we keep the structure of the gate closer to the PdB value, and the conductance gets closer to the PdB value. They do not affect the selectivity filter. Protonation decreases the conductance as we expected, and then addition of charged lipids slightly increases the conductance. The same happens with higher voltage simulations. So they result in slight changes in the selectivity filter and slightly higher conductance. And we also look at the pK values under voltage, and negative voltage slightly increases the conductance, even though the errors are pretty high. So I don't want to make definitive conclusions here. So just to acknowledge my colleagues of my student Ada Chen at Johns Hopkins. So she's here in the meeting. And then so she did, she's the first author on this more recent paper of on the conductance, and then an NIH, my long-term collaboration with Bernie Brooks, and then Dan Rowe, and at Erlang College, Bob Rosenberg. So yeah, so do you have any questions?
Okay. Thank you, Anna, for your very interesting presentation. I see already several raised hands. So Alexei, go ahead.
Hi, thank you. Very interesting. So I have 3 questions.
Okay.
So one is that, so what's the nature of selectivity for sodium? In other words, why is it doesn't conduct potassium, which is larger, or lithium, which is smaller?
Yeah,
just
that's
general.
Yeah, that's a great question, and we don't know the answer yet. So there are computational studies, well, we're actually working on it right now. So if you look at if you look at the conduction of sodium and potassium separately, or we're doing simulations also with mixed, we're getting very similar conduction rates for both. So I don't want to say too much, because we really require very long simulations to get enough sampling for this. So we are also looking at the polarizable force fields. There were some other people have also looked at selectivities, and again, it's really not clear at this point yet.
So for potassium is not just the simple size that's bigger, and this thing is the hole is tighter. That's not the case. It's not as simple as that.
Yeah.
Okay, so thank you. So my second question is technical. So you mentioned, I think, 16 or 11 total protonation states. I wonder why. I mean, given the symmetry, so you have for singly protonated, you have 1, and everything else rotational is symmetric, and for the doubly, you have diagonal across and next to each other. So it seems like you could get away with 3. As a physicist, I'm thinking that.
Yeah, no. So we did that in actually free energy perturbation.
Mm-hmm.
So that's exactly what we did, and we have a symmetry correction that then for free energy perturbation, we just looked at the S0 to 1s1, and from 1s1 to actually we two different S2s, because of symmetry.
Right.
You need those that are close to each other and diagonal ones. So yeah,
yes,
we do that. Mm-hmm.
But so why do something more complicated? I mean, it'll cost you in dynamics.
Well, the thing is, in EDS, you have to include all of the states in the Hamiltonian. So you can't really, you can jump from one to another. So you can allow that to happen.
So you cannot force this by symmetry, you cannot enforce symmetry and reduce the expense?
Correct.
Okay.
Correct. Yeah.
All right. And so my last question is, in general, so I think you're you're the perfect person to ask the question. And this is about about constant pH and their utility. Because you're not in the camp, in the camp that promotes constant pH, but you're using constant pH. So I wonder.
Wait.
Well, okay. So there are people who staunch proponents of the constant pH is the only way to go for any problem on Earth. But so you're using constant pH, but you're not in one of those camps. So I'm just curious, is that what I've been looking for is an example of a problem, clear example of a problem where you really just cannot, your answer would be qualitatively wrong if it were not for constant pH, for the coupling. So I wonder if this is one of those problems where if you considered separate simulations with those separate states, you would get a completely wrong answer, not just off somewhat, but a completely wrong answer. Again, I wonder if this is what's your hunch. Could that be the problem that really?
Yeah, that's what we initially thought. Especially because those protons could hop between different different glue residues, right? So, in the end, we got, when we did free energy perturbation, we were able to get similar results. So we did get similar results. So that was another thing that actually we wanted to check. If we did constant pH, do we get different results? And we got very similar results. So that's, okay. What I can say about this. Okay. But of course, you do want to, we didn't look at other ionizable residues, so I think it would be interesting to actually do that. So we just focused on the selectivity filter. It is in a way away from other residues, so we don't expect it to be coupled, but it would definitely be interesting to try, looking at many residues at a time.
Thank you.
Okay, Jim, you have a question?
Yeah, thanks, Walter, and thanks very much, Anna. So, this is going to be a silly question, so let me word it so it's not entitled, so I get somewhere. If you have a system of glutamic acid residues very close to each other, under what circumstances would they not protonate, but the conformation, but they would be sufficiently unrestricted, they would just come apart? Presumably that's not the case for this evolved channel, but the energies there are very, very large, with pK changes up by 10. And just in general, that's a biology question, not just for this system, but for other systems. You understand what I'm saying, so I'll let you answer that.
Yeah, but it does bind ions, and that's the thing. So ions very quickly, if you start the simulations with no ions, they quickly move in, so they don't like this huge negative charge. So, that.
The answer.
Yeah.
Thanks, Anna.
Mm-hmm.
I have a couple of questions, one more technical question. How did you impose the voltage on your simulation?
How did I impose what?
The voltage.
Oh, yeah. So we had electric field, so applied on all atoms, and then the ions redistribute themselves. So in the end, the electric field is felt across so the voltage drop is across the membrane, right?
Okay.
So this is standard what's done in these simulations.
So it's not really, so it's like an external fixed field rather than something that mimics an electrode, for example. So
yes, so we don't have the different membrane, different ion concentrations. Okay. There are some simulations where people do that, but they have to double the box size, because they need two sets of membranes to be able to maintain because of periodic boundary conditions. Yeah.
Okay. And the second question is, how did you calculate the local radius of the channel throughout the Z direction?
Yeah, so that was the program hole.
Okay, with hole.
Yeah. Mm-hmm.
Okay. Are there any? Yes, Guido, go ahead.
Very nice talk. I'm interested, is the evaluation of the conductivity something which is easy or not easy? I guess it's very noisy data.
Yeah, so you just count ions that are passing through. So you need to have enough events, and from that you get the current, and you can get the conductance. Yeah, so you really, what you need is really long simulations.
Right, and the voltage that you applied is comparable to the one that you have experimentally.
Yes. So we are mimicking the membrane potentials. Yes, so minus. Sorry, where is this?
Because this is like a non-equilibrium molecular dynamics simulation in a way.
Yeah. So you see, we have minus 100 millivolts, and then we also tried minus 400, and we do see that those are a little bit, they do bias a little bit. So minus 70 is your typical membrane potential in a neuron. So
Right.
really the minus 100 ones are the ones that are closest to nature. These are used, usually higher voltages are used to bias the simulations.
But when you have a higher voltages, you have higher error bars because you have fewer transition events?
This is just one. No, with the higher voltages, we have more transition events. So there are lots. Yeah, I don't see where else. Yeah, maybe this is a good one. So again, it's showing the errors in the conductance. So we do have more transition events, but we also have a lot at 100 millivolts.
Okay, thank you, very nice.
Mm-hmm.
Any more questions, comments? Okay, so let me thank again our speaker, Anna. Thank you very much for your presentation. So a little update. So starting next week, the speaker will be Wei Yang, and we come back to the usual day, which is Wednesday.